Our goal in this study was to identify new targets for therapy for leukemia. We used something called CRISPR screening, where we use genome editing to target every single gene in the genome in leukemia cells to look for genes that, when knocked out, cause the leukemia cells to die. What we found is that there are a subset of AMLs, or acute myeloid leukemias, that are dependent on a gene complex called the PI3 kinase gamma complex. And this was very interesting because PI3 kinase gamma is different from other PI3 kinases that are much more well-studied and targeted by therapies in cancer. The alpha, beta, and delta forms have actually drugs that are approved for various kinds of solid cancers and other leukemias, but gamma had not been studied. And what we found is that there's a subtype of AMLs that we can identify by particular biomarkers that seem more sensitive to inhibition of this PI3 kinase gamma pathway. And next, what's really exciting is that while I said this pathway has not been targeted in cancer itself, there are drugs that target the PI3 kinase gamma pathway that are being used in immuno-oncology as a way to stimulate the immune system in patients with solid cancers. So there are drugs, particularly one drug called eganelisib, which is a PI3 kinase gamma inhibitor that's already been tested in patients with solid tumors in combination with other chemotherapies. So what we were able to do was to take eganelisib then and test in the laboratory in AMLs, both from samples taken from patients and in models that we could test in the lab. And we could show that eganelisib, this PI3 kinase gamma inhibitor, worked in the subsets of leukemias that we could identify using these biomarkers. And so what does this mean for patients? It means that we can move very quickly to taking a drug that's already been used in other cancers to patients with AML. And so we are planning clinical trials, hopefully to start within the next year, where we will identify patients and be able to test this new therapy. But again, a therapy that we already know is safe and is well tolerated and is a pill, so an oral chemotherapy pill that we will be able to test in patients with AML, both as a single agent and in combination with established AML therapies.